Okay. All right. Yes. So we're finally going to get started. First of all, you can see that this educational program, we want to thank BioMarin for the support of this educational initiative, as well as CSL Behring, who provide funds for this ISTH Academy, especially with the discussion on gene therapy. And so, yeah, we're going to just go through a few of the currently approved gene therapies and discuss those, as well as a little bit of an eye to the future when we get to the end of the discussion. So here's the objectives. It's really to describe gene therapy correctly, including the basic terms, concepts, and then to look at the up-to-date gene therapy clinical data that we have from various clinical trials. It'll just be a brief overview of each of those. And then at the end, a little bit of an eye to the future. So here's where you can use your cards, your green for yes and red for no card. So do you routinely consider gene therapy as a potential treatment option for hemophilia patients who may benefit from it? So you can raise the green card for yes and the red card for no. All right, see pretty much all yeses, so that's great. Now, what is the goal of hemophilia treatment in general? So I like to think about it as the disease burden and the treatment burden. That's how I kind of categorize things. The disease burden really relates to essentially the bleeding. How does the bleeding complicate the life of the patient? How does the bleeding cause damage to the patient in terms of joints, muscles, intracranial? So basically disease burden and it is related to bleeding in hemophilia. And there's no treatment right now. As great as all these new treatments are, none of the treatments has gotten us down to zero bleeding. Ultimately, that's the goal, is to have really zero bleeds for all the patients. And then there's the treatment burden, which is the second part. That's the burden associated with actually doing the treatment. And we know with IV, with factor treatment, it's IV replacement, which is 1, 2, 3 times a week. Subcutaneous drugs, there's those that are daily, weekly, less often. But all of those require chronic, recurrent treatment that has to be remembered to be given and actually has to be given. And ultimately, the goal for me, as far as hemophilia, is to cure the disease. And, there's different definitions of what cure might mean. To me, it means that the factor level is put into the normal range and stays there indefinitely, or for life, really. And that would allow for this hemophilia-free life, where patients don't have to think about or worry about hemophilia. So gene therapy has brought us closer to that goal. I think by my definition of gene therapy, it hasn't brought us to that goal yet, but it's brought us closer to that goal. Because the one thing gene therapy is able to do that other treatments cannot do is offer a single-dose treatment that will ultimately, potentially reduce bleeding and, of course, not require other treatments, right? If it's a single-dose treatment, it doesn't require other treatments. And many patients who receive gene therapy do have essentially zero bleeds, as I'll show you. And if you have no bleeds and you're not having to infuse anything, either subcutaneously or intravenously, well, then that could be considered, as some people call, a functional cure. Essentially, you're living like as if you don't have hemophilia, even if your factor level may not be in the normal range. So on the surface, gene therapy is actually really simple. If you dive deep into it, it's very complicated. But on the surface, all we're trying to do is insert a healthy or functional copy of the factor VIII gene or the factor IX gene into a target cell that will then produce that protein, factor VIII or factor IX. So on the surface, it's actually quite a simple idea. Of course, executing that, as you probably are familiar and you've seen from clinical trials, the execution gets very, very complicated and can be quite challenging to understand all of the things that are happening. We don't really have a great understanding. But the bottom line is what we're trying to do, put the DNA into essentially what we have now is AAV vectors, deliver the gene to a target cell. The gene is in the cell and the proteins get produced. So these are the currently licensed gene therapies. I know they're not licensed in every country, but these are licensed in the US, Canada, and select European countries, and eventually we'll probably get to other places in the world. So we have for hemophilia A, valoctocogene roxaparvovec, and you see the structure of that factor VIII gene. I'm not going to get into the details other than to say it's a B domain deleted factor VIII, and that may or may not have some implications on the production of protein. And for hemophilia B, there's two. There's a tranacogene dezaparvovec, which uses a Padua mutation, which is what's indicated in the red, to enhance the production, the activity of factor IX from the factor IX that is synthesized. So this is a high functional variant of factor IX. And then fidanacogene elaparvovec was just recently licensed, and it also uses, they use a different term there. You see human factor IX R338L variant. That's really the Padua variant, just like what it says for a tranacogene dezaparvovec, AGG to CTG. So those are the structures of those three, and they all use an adeno-associated virus to deliver the gene to hepatocytes, which is the target cell of all these gene therapies. So let's take a look at valoctocogene roxaparvovec. This is from the phase 3 GENERATE-1 trial. This was the pivotal phase 3 trial published in the New England Journal of Medicine. What I'm showing you here, and that's why the reference is a bit different, is this is the 4-year data that was presented just a few months ago. This is basically for adults with severe hemophilia A. They all had to be on prior factor VIII prophylaxis to enter the trial. They could not have factor VIII inhibitors, and they could not have antibodies to the vector, in this case AAV5. And they also had to have a healthy liver. The endpoints are factor VIII activity, and then changes from baseline during the post-infusion phase, or post-prophylaxis to factor VIII phase, of bleeding rates, factor VIII infusion usage, and health-related quality of life, and of course safety. So here we're looking at factor VIII levels on the left side. You're looking at year 1, 2, 3, and 4. Well, let's focus on year 4 because year 1 is in the past, so is year 2 and 3. In year 4, you'll notice in gray is those patients who are above 40 units per deciliter or 40%, meaning not in the hemophilic range. And in blue is 5 to 40%, which is the mild range. And if we add those two together, that's 60% of the patients who are in the mild hemophilia range at least. And then a smaller portion in the moderate range and some that are below the limit of detection. On the right side is the annual factor VIII infusion rate, and this is the number of infusions per year. In the baseline, it's around 130. That's because some patients were on extended half-life factor IX, so about 100 infusions a year. Others were on standard half-life, so about 150 infusions a year, so the average was around 130. And you'll see that after their infusion, year 1, 2, 3, and 4, that goes down pretty close to zero. It's a 92% reduction, particularly focused on year 4. So substantial reduction in the use of factor VIII. So remember, the goal of gene therapy is to provide a factor VIII level that is therapeutic, that will prevent bleeding, and that will not require factor VIII infusions. So here I show you the factor VIII level. Here I show you the reduction in infusions. What I didn't have time to show you is the bleed rates, but those go down commensurately as well, and patients have relatively low bleed rates. In fact, most of the patients are not having any bleeds at all. Let's talk about a tranacogene dezaparvovec. This is for factor IX. So again, the study design, all of these have a very similar study design. A lead-in phase while on factor IX, in this case it's factor IX prophylaxis. And again, the eligibility here is adults with severe hemophilia B or actually moderately severe hemophilia B, so up to 2% factor IX. They had to previously have factor IX, be on factor IX prophylaxis, no inhibitors. Now, in this case, patients with pre-existing AAV antibodies to the vector, were tested, but even if they tested positive, they could be enrolled. So that's different than what happened with valoctocogene roxaparvovec, where those who tested positive were excluded. Also had to have a healthy liver, and the endpoints are basically the same as the endpoints for all the trials, factor activity, and changes in bleed rates and infusion rates. So unfortunately, the data that I'm presenting here is in a just slightly different version. The last one was like a bar chart, and this is like a donut. But let's walk through it. So in the yellow color is factor IX levels that are in the normal range, 40 to at least up to 100%, or at least normal to near normal range. you see a third of the patients are in the normal to near normal range, and this is at year 3. So it's a bit different than what you saw for factor VIII. And here we divide the mild hemophilia group into 2 groups. What I like to call mild hemophilia, so above 12%, you'll see 12 to 40%. And then the lower range of mild from 5 to 12%. The mild, so 12 to 40% is 48%, so almost half the patients. And then a smaller group of 5% are in the lower mild range. And then a few patients are below that range, as you can see. Essentially, over 87% of the patients were at least in the mild and normal factor IX ranges here. What about reduction in factor IX usage? So this is just a different scale. The scale here is in units per year as opposed to number of infusions per year. But essentially, it's showing you really the same idea, where in the lead-in phase, patients were receiving 250,000 units per year of factor IX, which is the prophylaxis dose typical for an adult. And you can see a 96% reduction, very similar to what we saw for valoctocogene roxaparvovec. So after the infusion, patients are just not needing to infuse factor IX or very, very little. And almost half the patients, as you see in the blue bar, 46%, did not require any factor IX infusions for at least 3 years since they were infused, right? This is 3-year data. As far as safety, there were 11 participants who had ALT rises. Some were treated with corticosteroids for about 3 to 4 months. No late ALT elevations were noted, and there were not other significant safety issues. And then finally, fidanacogene elaparvovec, the most recently approved molecule. I guess this looks familiar by now. I mean, the trial designs are basically the same. Now, one difference between etranacogene dezaparvovec and fidanacogene elaparvovec is, in this case, if you tested positive for antibodies against the vector, you could not receive this. You were excluded. That's one differentiator between the 2 factor IX products. Is one, if you test positive, you can still get dosed, and the other one, you cannot get dosed. I guess was briefly on the slide. Let me just say that in the etranacogene dezaparvovec, about a third of the patients had positive antibodies, and all but one of those did have a factor IX level that was, generally speaking, similar to the patients without antibodies. There was one who had a very high titer antibody. It was over 3,000. The next highest was around 680. That one did not have a response. But all the others who tested positive for etranacogene dezaparvovec did have a response. In this case, fidanacogene elaparvovec, these patients, if they tested positive for the vector, for antibodies to the vector, they could not receive the product. They were just excluded. So that's another difference. Otherwise, same inclusion criteria, same endpoints, basically same trial design. So here's the result. This is looking at a specific one-stage clotting assay. You're looking at the factor IX activity. Here the scale goes out to 104 weeks, so 2 years. Because this trial started a bit later. And you can see that we've got about 10% of the patients that are in the normal range, about another 50% that are in the mild or upper mild hemophilia range. Now notice here it's 15 to 40, not 12 to 40. It's kind of annoying. All the trials have slight differences, but this is fairly similar in terms of the breakdown. And then 5 to 15, and then a smaller portion, about 20%, that were less than 5%. So about 80% of the patients here had levels at least in the mild range. With etranacogene dezaparvovec, it was about 87%. So pretty close. Again, the patient populations might be a little bit different, so I wouldn't take those percent differences to be all that meaningful. Here's the infusion rate drop. Again, very similar, 92%. So it was 92% for valoctocogene roxaparvovec in terms of reductions in infusions after the gene therapy. For etranacogene dezaparvovec, it was 96%, and here it's 92%. Basically all in the same range, substantial reductions in factor usage, with most of the patients not needing, you see in this case, 64% of participants needed no infusions after week 12 through month 15. And in this case, 13% resumed factor IX prophylaxis. So 87% are still off of factor IX prophylaxis. As far as safety, there was a more substantial proportion of patients here who had high ALT levels and ended up on corticosteroids, but none of the patients needed corticosteroids beyond a year. So there's some differences in the corticosteroid use and ALT elevations between these two factor IX products, but at the end of the day, eventually, all the patients are no longer needing the corticosteroids. All right, so here's a question for you with the cards. Do you consider potential immune responses to viral vectors used in hemophilia gene therapy a significant barrier to its success? So yes or no, we've got a mixture of yeses and nos. Looks like a little bit more on the yeses. Yeah, I mean, I think it depends on how you define barrier to success. I mean, it is a bit of a barrier in the sense that patients may need to go on steroids and have to get tested. But I think I would view it also as, in the long run, for all three of these molecules, by 1 year, almost all the patients for all three were off steroids by a year. And most of them substantially less than that. So keep that in mind, that the corticosteroid needs were fairly early on and not long lasting. Now, the question is, how much of that impact the ultimate factor levels? And that's another question to be thinking about as we move forward in time. So what about these neutralizing antibodies to the vector? So this is just a schematic showing the general incidence of these vectors. So you see vectors, 3 different vectors, AAV2, AAV5, and AAV8. And it shows about a 40% prevalence of these pre-existing antibodies in hemophilia A and hemophilia B. I already mentioned, and you can see in the blue on the bottom, we did discuss this, that in patients with pre-existing antibodies to AAV5 that were included in the trials, there was the one patient who was 4.5 times higher than the others who didn't respond, but all the other ones did have a response. And for Hem A, patients were excluded. So I just want to close a little bit about the limitations of this current approach. And at this meeting, we've already heard some interesting, and there's more, discussions about the next sort of iterations of gene therapy, kind of beyond AAV. And I was just in one that showed, using essentially an ultrasound bubble technique to get DNA into the cells. It was kind of very interesting. And there's other stuff out there. But I guess the question is, what are the limitations of AAV gene therapy? Well, many individuals have pre-existing antibodies. So AAV is a ubiquitous virus. Many people have been exposed to it. Now, it doesn't make humans sick, so you'd never know if you had it. But if you have the pre-existing antibodies, it either excludes you. In fact, for most of the drugs, 2 of the 3 drugs that are approved, you're excluded. You have to actually get tested before, in order to, in the commercial setting, you have to get a FDA-approved test to determine whether or not you have the antibodies. And if you test positive, you can't get those molecules. For tranacogene dezaparvovec, it's recommended to get tested, and then depending on the result, the treating physician may or may not still treat the patient. High vector genome doses are needed, which could result in some toxic effects. We haven't seen that in hemophilia so much, but in other diseases, when you start to push the dose, you start to see people dying, actually. But we don't use those doses in hemophilia, and we have not seen deaths from gene therapy in hemophilia related to the toxicity of the gene therapy or the vectors. Immune reactions against the capsid, which is the ALT elevations, high degree of variability in responses. We do get quite a lot of variability in the responses. The hepatotoxicity, long-term, doesn't seem to be a problem, but it's still something we have to be looking out for. And this approach is likely not going to be effective in children who have rapidly dividing cells. And the AAV is a non-integrating vector, so the vector is not integrated into the DNA. So as cells divide, that DNA doesn't get copied. So the daughter cells don't get the copy. So as a cell divides once and then twice, you get a dilution of that DNA because it's not integrated into the chromosome, so it doesn't get duplicated when cells divide. And of course, young children have a small liver, maybe the size of my fist, and then an adult, you need pretty much 2 hands to hold their liver. So it grows a lot. Clearly, there's been at least added cells, if not all new cells. So are you involved in collaborative efforts or research initiatives aimed at advancing gene therapy techniques for hemophilia treatment? Are you guys involved in any of that? Most of you, I see no's, okay? Some yeses, but mostly no's. So how do we overcome these limitations? So as far as pre-existing antibodies to the vector, well, we have to think about using a non-viral method to transmit the DNA. And there's some work being done with non-viral transduction, either with lipid nanoparticles or actually this very interesting technique I saw this morning using essentially IV ultrasound and getting the DNA into the cells that way. It really didn't make much sense to me when I had heard that that could be done, but the presentation showed that it could. So basically trying to use non-viral vectors, because if we use viral vectors, there's always the possibility of having these pre-existing antibodies. High vector genome doses, if we can get the doses lower, that may be less toxic, and also you could produce a lot more and make it possibly a lot less expensive. So using novel techniques such as integrating the functional gene with gene editing, and there's a trial that is starting looking at for hemophilia B that is using CRISPR-Cas9 gene editing to actually put the factor IX gene directly into the chromosomal DNA. Immune reactions to the vector, again, that's the immune system reacting after, not pre-existing antibodies, again, using non-viral vectors. High degree of variability, well, obviously the obvious answer is to use systems with a more predictable response. And the inability to treat children, we'd have to use integrating vectors such as lentiviruses or gene editing. So with that, I'm going to stop. There's not really a Q&A here, but I can pop down here and take a few questions for people who have them. Oh, there is Q&A. Oh, so never mind. Okay. They have a microphone for you. Sorry, it's so loud in here. I don't know how you do a Q&A. But yeah, if you have any questions, please go ahead and do that. And then you can evaluate this presentation. Trust me, I will not see the results. So if you want to all give it a thumbs down, it's up to you.
If you're using the lentiviral vectors, the package site will be enough for the gene?
Yeah, yes, there have been some, I'm not hearing my own answer. There's been some, there's work with lentiviruses, and yes, they are big enough to package both factor VIII and factor IX. With factor VIII, it's B domain deleted, and the factor IX gene is fairly small.
Thank you.
All right. Well, thanks, everybody.